half reaction. All right? So if we separated these two into their two separate half reactions, so for the oxidation, we see glucose being oxidized to CO2, and we get the production of 24 electrons. And then the reduction half reaction for this process would be oxygen combining with the 24 electrons to produce water. Okay? So oxidation reduction. So for this oxidation half reaction, what is this? What is this entire process that we have been looking at? When we take glucose, we oxidize it to CO2, produce a bunch of electrons in the process. What is that we have already talked about thus far? Citric acid cycle and glycolysis, right? So we have dealt with the oxidation part of this. This is glycolysis as well as the citric acid cycle. In the process, we have formed 24 electrons, right? So just as a reminder, our little electron carriers, NADH, how many electrons are transferred to NADH in each reaction? How many electrons does it carry? 2, right? So NADH transfers its electrons in the form of a hydride. So a hydride is hydrogen with 2 electrons, right? It has a negative overall charge. So NADHs are 2 electron carriers. We see the exact same thing for FADH2. These are both 2 electron carriers. So we take glucose and we form 24 electrons in its complete oxidation, then how many electron carriers do we form if each carrier contains 2 electrons? Reverse. 12, right? 12. We get 12 separate electron carriers. And how do we know those as at this point in time, right? We just went over the summarization of all the NADHs and all the FADHs that we formed in glycolysis in the citric acid cycle. So we have 12 electron carriers. How many of those are NADH at this point in time? So 10 of them are NADH, and then 2 of them are FADH2. Again, each of these guys contains 2 electrons, so this is our 24 total electrons, or 12 electron pairs. Okay, so this is what we have. We've got our 24 electrons in the form of these electron carriers. Now, this chapter is going to deal with the reduction half reaction, right? How do we take these electrons and reduce oxygen to water? Most importantly, when we do that, we're going to generate a bunch of energy. How are we going to utilize that energy generation and couple it to the production of ATP, which is the whole long-term goal of this, okay? So we're going to talk about, number 1, how we take the energy that is present in these electrons, these electron carriers, and reduce oxygen to water, and then we'll talk about how that is coupled overall to the synthesis of ATP. Okay, so oxidation reduction. First, before we can do that, we need to talk about where all of this occurs. So I've been kind of bad about that. I don't like to talk about cell biology because it makes me nervous in life. But we need to talk about that because it's kind of important in the overall architecture of how the electron transport chain is laid out, okay? So there are 2 different types of organisms in the world, right? What types of organisms? There's eukaryotes and there's prokaryotes, okay? Eukaryotes, which is what we are going to focus on, have a mitochondria, okay? So eukaryotes have a mitochondria. And the mitochondria is the location of all of the cool happenings that are going to take place in this chapter, as well as last chapter with respect to the citric acid cycle, okay? So what happens in the mitochondria? Well, number 1, we have our pyruvate dehydrogenase complex. We have all of the enzymes of the citric acid cycle. We will also have the enzymes of fatty acid oxidation, the electron transport proteins, as well as our ATP synthase. So there's lots of stuff taking place in the mitochondria. What is not in the mitochondria? What is instead in the cytosol? Right, so glycolysis is the only poor little guy left out of all of the fun stuff that happens in the mitochondria. Okay, so let's talk about a couple different components of the mitochondria itself. So the mitochondria has what is called an outer membrane. This is a nice smooth membrane. So we have this nice little outer membrane here. I'm going to abbreviate this OM. It also has what is called an inner membrane, right? So this inner membrane is on the inside. I'm going to, again, abbreviate this IM. Most importantly, this inner membrane is not smooth, right? Instead, it is filled with things called cristae, or these invaginations or folds, right? It's not the smooth surface that you get with respect to the outer membrane, okay? So we get these nice little folds or invaginations in the inner membrane. What does that do overall? Just going back to what you guys know. Increases surface area, okay? All of the enzymes of the electron transport chain are going to be in that inner membrane, right? If we increase the surface area of the inner membrane, then we increase the amount of enzymes or electron transport proteins, and we increase our overall net rate of respiration. Okay, inner membrane, outer membrane. We also have the inside of the mitochondria. What is this called? Inside the inner membrane is the mitochondrial matrix. And there's also a space in between the inner membrane and the outer membrane. What is this called? Intermembrane space, right? So I'm going to call this guy IMS, okay? So the matrix is going to have all of our soluble enzymes, all of our soluble various cofactors, etc., with respect to what happens in the mitochondria. The inner membrane is going to have all of our proteins that we're going to be interested in. Okay, so all of our electron transport proteins, I should say. That's more important. Okay, so a couple more things. What's the difference between the outer membrane and the inner membrane besides the presence of the folds or invaginations? So number one, the outer membrane contains things called porins. Okay, so porins are these little small proteins that essentially allow the free diffusion of molecules that are smaller than 10 kilodaltons. Okay, so that's going to allow the free diffusion of pretty much everything besides enzymes. Okay, what that's going to do overall is it's going to make this inner membrane space have the same composition as the cytosol, because the presence of these porins again allows free diffusion. So the cytosol in the inner membrane space are going to essentially be at equilibrium. They're going to have the same constituents. Okay. On the other hand, the inner membrane does not contain porins. Okay, so it is not freely diffusible. The only thing that is freely diffusible in the inner membrane are small molecules like oxygen, CO2, and water. Any type of ions is going to have to go through a transport protein, as well as any type of cofactors like ADP, ATP, etc. Those are all going to have to go through transport proteins to get into the matrix. Okay, that's because, again, they do not have any type of porins on the inner membrane. Another thing to note about the inner membrane is that more than 70% of the inner membrane is protein. That leaves approximately 30% lipids with respect to the inner membrane. Comparatively to all of the membranes in the body, this is the membrane that has the most amount of protein embedded in it. Okay? And of that 70% protein in the inner membrane, about a half of those are due to the proteins of the electron transport chain and oxidative phosphorylation, whereas the other half make up these transport proteins that allow the diffusion of small molecules like ADP, ATP, as well as ions in. So these guys are transport proteins. Okay, so that is the scene of everything that's going to happen. Okay, so now, a couple of things. What are these proteins or these complexes of the electron transport chain? There are 5 total complexes of the electron transport chain. They are labeled complex 1 through 5. It's very clever, okay? For the electron transport chain itself, complex 1 through 4 is what makes up the proteins of the electron transport chain. Transport chain. What is complex 5 then? So complex 5 is our nice little ATP synthase, which we'll talk about at the end. Okay, so ATP synthase is how we're going to essentially make ATP. The proteins of the electron transport chain itself, which are going to take NAD long term and reduce oxygen, are complex 1 through 4. Okay? Okay, so now let us consider this overall reduction of oxygen from NADH. So we have our nice little electron carriers. And what's going to happen with respect to the electron transfer chain is we're going to enter our little electron carriers in the form of NADH. These electrons, long term, are going to reduce oxygen to water. Okay, right? That's the big whole purpose of this. This is going to generate a bunch of energy, right? How would we measure how much energy it generated? What would be a nice little helpful means to do that, knowing that this is a redox process, and all of these guys have standard reduction potentials? So we can look at the various reduction potentials between these two half reactions. Okay, so the half reaction for the reduction of NAD plus to NADH gives us a standard reduction potential of a minus 3.15 volts, and the half reaction for the reduction of oxygen to water gives us a standard reduction potential of a 0.815. Okay, knowing what you guys know about reduction potentials, how do electrons like to move with respect to reduction potentials? Do they like to go from high potential to low potential, or do they like to go from low potential, negative potentials, to high potentials? So reduction potential is not in the terms of actual chemical potential. What is the tendency going to be? Are we going to have NADH reducing oxygen, or are we going to have water reducing NAD+? So the tendency is always the transfer of electrons that are at low potential, negative reduction potentials, to things that lie above it, that are at high potentials, or positive reduction potentials, right? How can we measure the potential difference between these two half reactions? We could calculate a nice little cell potential, right? The cell that is formed by these two half reactions. How do I do that? I take the reduction potential of what and subtract the reduction potential of what? Right, so I take the reduction potential of the thing that gets reduced, right, the cathode, and I subtract from it the reduction potential of the thing that gets oxidized. So I could do that to calculate the overall cell potential that is generated in this half reaction. So who gets reduced here? Oxygen gets reduced, so we've got our reduction potential for the reduction of oxygen to water, and we're going to subtract from that the reduction potential for NADH. Overall, we get a nice positive 1.13 volts, right? Is this a spontaneous process? We have a positive cell potential. Maybe I should have gone back over electrochemistry, right? So if it is positive, yes, we do have a spontaneous process. So positive cell potentials give us overall spontaneous processes. What reaction or what equation do I utilize to take my cell potential and turn it into Gibbs free energy, which is what we're more familiar with? So delta G is equal to a minus, this is where the positive and negative come in, minus NFE, right? So positive E's give us negative delta G's overall because of the negative in front. What does N refer to? Number of electrons, and for this process, how much is that? Right, we have 2 electrons being transferred. F is just some constant, who cares about it, 485, and then E is our plus 1.3 volts. So overall, with respect to this reaction, which occurs in your body every day, this is capable of generating a minus 220 kilojoules per mole. Okay, so that is the overall amount of energy that the reduction of oxygen via NADH could generate, right? I've also told you already that for every 1 NADH, we only generate about 2.5 ATP in this process of oxidative phosphorylation. How much approximately is an ATP cost to phosphorylate it from ADP and PI? Like 30-ish kilojoules, right? So it's a positive, it's an exergonic reaction. So for every 1, I get 2.5. So the cost of making 2.5 ATPs would essentially be 2.5 times approximately 30, and I get the approximately look like a negative, that's bad, so I'll just say 30. So approximately this ends up being about 80 kilojoules per mole that we actually need in the synthesis of these 2.5 ATPs. So there's lots more energy available than what we actually use, okay? Why would that be the case? The efficiency of this is only like 30, 35%. In reality, it ends up being like 60 to 70, right, with the actual conditions in vivo. But why would you want to waste so much energy? This kind of goes back to the idea behind reversibility in chemical reactions. If you throw away a lot of energy in the form of heat, is the reaction reversible? No, right? And so we waste a lot of energy in this process, but that's okay because it makes the reaction completely irreversible, right? You're never going to see ATP go back through ATP synthase and somehow, I guess, do the reverse of this. That's what I'm trying to get at. Okay. All right. So again, we waste lots of energy in this, but we do it for a good cause. It's to make sure that this is an irreversible process. Okay, so overall, electron transport chain. There are 4 separate complexes of the electron transport chain, right? Yes. What did I do? What did it say? 30. Oh, 30. That's like, so to take ADP and combine it with inorganic phosphate to make ATP, that's about what it costs, about 30 kilojoules per mole. I should put that. This is a, oh no. From, again, just the phosphorylation of ATP to form ADP. Hey, nope, yep, who cares? Okay, all right, so here we go, right? We've got 4 different complexes. What's going to happen, right, if we just took NADH and we combined it with oxygen to form water, you get this nice big reaction, right? It releases beautiful 220 kilojoules per mole of energy, and that would be lost in the form of heat. Okay? So what we do with respect to biology, and this is why the electron transport chain is arranged the way it is we essentially put NADH and oxygen in a series of 3 separate electron transport complexes. Okay? By doing that, we break up the energy of that NADH going to oxygen to form water into 3 separate parcels. And in each of those parcels, we're going to couple the energy that's generated to the translocation of protons from the matrix to the intermembrane space. Okay? So we did this all at once. If we just mix some NADH with some oxygen, it would react, but we would lose all of that energy in the process. Okay? By separating it into these 3 or 4 different complexes, we're going to break up the energy that's present into 3 to 4 different parcels, and in each of those parcels, we're going to utilize the energy to translocate protons from the matrix to the intermembrane space, which will be utilized at a later date. Okay? That is the overview of the electron transport chain. Again, there are 4 complexes. Okay? Only 3 of those are actually going to participate in the translocation of protons from the matrix to the intermembrane space. Okay? That ends up being complex 1, complex 2, and complex 3, and complex 4. Okay? And we're going to talk about what each of these complexes does right now. Why not? Let's talk about it right now. Okay, so complex 1, what we're going to see happen is we're going to bring in our little NADHs. And what complex 1 is going to do is it's going to take the NADH and it's going to transfer its electrons down this nice little chain of redox cofactors. And at the end, it's going to reduce something called coenzyme Q, producing reduced coenzyme Q. There are a lot of different names for coenzyme Q. And we're going to talk about them when we actually talk about these redox cofactors. But long term, complex 1 takes NADH and we produce reduced coenzyme Q. Okay? Complex 2 is going to take our other little cofactor of the carboxylic acid cycle, so FADH2, and it is also going to take those electrons and it's going to reduce coenzyme Q with it, forming reduced coenzyme Q. I wish I could call it its other name, but I haven't gotten there yet. All right, so now, complex 1, lots of energy in this, right? NADH is a very low potential, coenzyme Q is right above it. So in the process of transferring its electrons on this nice little redox chain, we are going to generate enough energy to pump protons, right? We get a nice positive reduction potential. We generate a lot of energy from that. We're going to get proton pumping. FADH, on the other hand, FADH2, does not have such a low reduction potential. So when we transfer the electrons from FADH2 to coenzyme Q, it's going to be spontaneous, but it is not spontaneous or energetic enough to translocate protons. Okay? So the only purpose of complex 2 is essentially to get the electrons that are stored in our FADH2 into the pool for later reduction of stuff. Okay? So we're not going to see any type of translocation of protons in complex 2. Now, what happens in complex 3? So in complex 3, we take our little reduced coenzyme Qs, and we transfer electrons down this nice little redox chain until they get to cytochrome C. Okay, so take electrons from coenzyme Q, we reduce cytochrome C. Complex 3 is going to have enough energy in this process of reduction of cytochrome C to translocate protons, right? And then finally, what's going to happen in complex 4 is we're going to take our reduced cytochrome Cs, and they are going to react at our little complex 4, or cytochrome C oxidase, and they are going to reduce oxygen to water. There's going to be lots of energy available in this guy, and so we also are going to get translocation of protons. Okay, so now that's kind of just the overview, unfortunately. Let's briefly talk about some of these cofactors that I have totally neglected to talk about at this point in time. If we go over to this table, we will find that the electrons don't just go from NADH to coenzyme Q. Instead, what they do is they follow this nice little path down a bunch of redox active cofactors, right? And these redox active cofactors are things like these iron sulfur clusters and these flavins and these hemes. Okay, so we're going to talk a little bit about these cofactors so that we can help understand the reactions that take place at each of these little individual steps. Okay, so first we are going to talk about iron sulfur clusters, which we kind of have mentioned before. We're going to talk about flavins a little bit, and then we're going to talk about coenzyme Q. Okay, we're going to start by talking about flavins. So we have talked about flavins already, right? So flavins we talked about with respect to flavin adenine dinucleotide, FAD. The cofactor that we see besides FAD in the electron transport chain is something called FMN, or flavin mononucleotide. Okay, so what's the big idea behind flavins? What's so great about them comparatively? So remember that our NADHs are 2 electron transport, or 2 electron carriers, right? NADH can only transfer 2 electrons at a time. It can give it away or it can accept it, right? But it has to be 2 electrons. I mentioned something about flavins last time that was unique to flavins comparatively to NADHs. So flavins do hold 2 electrons. They can be 2 electron reduced, but can they also just accept 1 electron at a time? Yes, right? And so flavins, what's good about them at least, is the fact that they can accept 2 electrons, or they can also just accept 1 electron at a time. So these guys can accept 2 electrons or 1 electron. And the reason for that is because the semiquinone form, or the radical form, which is what you get upon 1 electron oxidation, is very stable, right? So it's okay taking 1 electron, forming this radical, and just being that radical for a while, okay? So we can undergo a 1 electron transfer to produce the semiquinone form, and then we can also reduce that even further to the fully reduced form, or the hydroquinone form. Okay, so coenzyme Q is the exact same way, okay? So coenzyme Q, I'm going to talk about it for one little second. So coenzyme Q has a moiety that is called a benzoquinone, okay? So this is a benzoquinone. This benzoquinone is attached to a long hydrophobic isoprene tail, okay? Overall, what this does for coenzyme Q is makes it extremely hydrophobic, so much so that you can call this lipophilic. I'm not good with the fats. I probably should work on that. Okay, so lipophilic. What does that mean with respect to coenzyme Q and its location of habitation? It means that this guy essentially resides in the actual membrane itself. Okay, so the lipid bilayer, this is where our little coenzyme Q resides. It is not soluble. It is membrane soluble. A couple other things, because I know I'm going to get started with it, and I'm not going to stop. There are other names for coenzyme Q, right? So one of them is called ubiquinone. The reason why they call it ubiquinone is because it's everywhere, right? It's ubiquitous, so it's called ubiquinone. So I will probably start referring to coenzyme Q as ubiquinone, okay? If I do, ubiquinone is the oxidized version, ubiquinol is the fully reduced version. So I'll tend to start saying ubiquinone, that's the oxidized form, ubiquinol is the reduced form. And again, great thing about coenzyme Q, just like flavin, it is also a 1 or 2 electron transport cofactor, right? So it is also stable after 1 electron reduction in the semiquinone form, and then it can also gain another electron to form the fully reduced form. Oh gosh, this took a while. Okay, so then, what else? Iron-sulfur clusters, right? We've talked about iron-sulfur clusters, I think-ish, maybe. There are going to be a lot of them in the electron transport chain, and they're going to take on 2 main forms. The first form is something called a 2-iron 2-sulfur cluster, and the second is a 4-iron 4-sulfur cluster. So what's the big idea behind these iron-sulfur clusters? So number one, they have iron ions, right? So we've got iron ions, and the number of irons is given by the 2 in front. So this is a 2-iron 2-sulfur cluster. It has 2 irons in it. These irons are coordinate or form a coordinate bond with, first off, the thiol groups on cysteines in the protein itself. So that coordinates via 1 ligand, right? Also in these iron-sulfur clusters, they are also coordinated by something called inorganic sulfide or acid labile sulfide. Okay, so these are actually sulfide ions, so S2-, that also form coordinate bonds with these irons. Okay, we see the exact same thing in a 4-iron 4-sulfur cluster, except in a 4-iron 4-sulfur cluster, we get 4 irons that are bound with sulfides, 4 sulfides. That's where the 4-iron 4-sulfur comes from. These guys are also attached to the protein via cysteine thiolates. Regardless of the iron-sulfur cluster, iron is in the tetrahedral molecular geometry in both of them. It forms a tetrahedral molecular geometry. Most important, it moves between an iron 2+ oxidation state and an iron 3+ oxidation state. Okay? So even though there are multiple irons in these cofactors, it only essentially goes through 1 oxidation state. So these guys form this kind of conjugated system where they are only capable of transferring 1 electron at a time. Okay, so it goes from a 2+ to a 3+. Most important, 1 electron transfer proteins, or cofactors. Okay, iron-sulfur clusters. Last but not least, something that we have also already talked about before, are hemes, right? We've talked about hemes. Where have we talked about hemes before? Myoglobin, hemoglobin, right? So myoglobin and hemoglobin, what type of heme did it bind? It bound something called heme B, right? This was an iron protoporphyrin 9, okay? So hemes are iron-containing proteins, right? They are coordinated by the nitrogens that are present on these little porphyrin rings, okay? Comparatively to the irons that were in the iron-sulfur clusters, those were in a tetrahedral molecular geometry. What is the geometry of iron in these heme groups? You'll have 4 ligands that are in the plane of the porphyrin. What else is there too, though? There's usually 2 other ligands that did the 2 coordination sites that are above and below. So overall, what type of geometry would that form? So we've got 4 in the same plane and then 1 above and 1 below. This would be octahedral. So comparatively to the irons that are in the iron-sulfur cluster, that's tetrahedral. These are irons that are in the octahedral geometry. Couple of things. There are different types of hemes depending on the heme that is actually, or the various constituents off of the heme. We've talked about heme B, all right? So heme B, there it is, protoporphyrin 9. It's got an iron attached to it. There is also a version of heme called heme A. The only difference between heme A and heme B is the fact that heme A has this very long hydrophobic isoprene group at the top, right? So what that's going to do to heme A is make it a little bit more hydrophobic towards the interior of the cofactor. There's also another type of heme called heme C. The only difference between heme A, heme B, and heme C is the fact that heme A is actually covalently attached to the protein by the formation of these thiol bonds with the vinyl groups that are on the heme, okay? So heme C, we've got a covalent attachment. Heme B, it's non-covalent, but it's in the protein. And heme A, same thing, but we've got this nice hydrophobic tail attached to it. Okay, so that's great. All of these hemes are also going to participate in one electron transfer processes. So a heme can only move between an iron 2+ oxidation state and an iron 3+ oxidation state. So because of that, these guys can only transfer one electron at a time. There will be an exception to that, and I will mention it when we get there. Okay, so one more thing before I stop. If we go back to that slide that I showed you, oh, here's all the reduction potentials of all the redox cofactors in these complexes, you should have noticed, ideally, that there were a lot of iron-sulfur clusters. There were a lot of hemes, right? But they all had different reduction potentials, even though they're the exact same cofactor themselves. Why do you think that could be possible? How could that be possible? So all of these guys, even though they're going to be duplicated ad nauseum through all of these redox cofactors or all these electron transport chains, all have different reduction potentials, right? So what could be modulating the reduction potential of these various cofactors? So you could kind of consider it, if we go back to the idea behind pKa of ionizable side chains, was a pKa of something like lysine, was it always going to be constant at that exact same pKa that it was at when we had the free amino acid? No, right? What was going to modulate the pKa? The environment that it was around, right? The microenvironment that it was around. If it was around, let's say we take lysine, if it was around a negatively charged aspartate or glutamate, right? It was going to make it easier to protonate that lysine because it had a negative charge. It would also like to be positively charged. So what would that have done to the pKa of that lysine if we wanted to make it easier to protonate? It would have decreased the pKa, right? So again, the various factors that are in the protein, the microenvironments that are in the protein, are going to be affecting the various redox properties of these cofactors. So even though they're the exact same iron-sulfur cluster, because of the different microenvironments around them, they're going to essentially have different reduction potentials. And that's why we see this nice little beautiful chain going from very negative reduction potentials to very positive reduction potentials as we move through our complexes. Okay, so I'm going to stop there, and then we will start again talking about complex 1. Proton, or to the synthesis of ATP, okay? Okay, so as a reminder, we have NADH, right? So NADH is going to transfer its electrons long-term to oxygen, right? So we transfer electrons to oxygen, we're going to get the complete reduction of oxygen forming water, okay? So that's one source of electrons into our electron transfer chain. What is another source? Where else are we getting electrons from? FADH2, right? So another source of electrons that are coming from the citric acid cycle is FADH2. Again, we're going to be transferring these electrons into our electron transport chain to generate water, okay? All right, so the first step in the electron transport chain is called complex 1, okay? So all of these complexes of the electron transport chain also have other names, okay? So the other name for complex 1 is this NADH coenzyme Q oxidoreductase, okay? Now, overall, what is going to happen at complex 1 is we're going to have NADH. NADH is going to transfer its electrons down this little electron transport chain in complex 1, and at the end of complex 1, it is going to reduce coenzyme Q to reduce coenzyme Q, right? What was another name for coenzyme Q? Ubiquinone, right? So when we reduce ubiquinone, we produce ubiquinol, okay? So this is going to be QH2. All right, so now, overall structure of complex 1 is gigantic, okay? So with respect to the eukaryotic version, it is one of the largest electron transport complexes in the overall mitochondria. So this is the biggest overall protein complex, okay? In it, there are 14 core subunits, okay? So depending on the organism, is going to depend on how many actual subunits there are, but regardless, there are 14 core ones that make up all of them, okay? So 14 core subunits with respect to complex 1. Okay, so in the eukaryotic version, or the mammalian version, there are actually 30 other subunits that go on to these 14 core subunits, okay? These are just going to be extra subunits that are going to help in stability as well as regulation overall, okay? But again, I'm going to talk about this with respect to the 14 core subunits. All right, so we have the crystal structure from complex 1 from the bacteria Thermus thermophilus, okay? And as a note here, I want to make a note of this. With respect to all of these complexes of the electron transport chain, these are all integral membrane proteins, okay? Meaning that not only are they large, gigantic complexes, but they are also embedded into our membrane, right? What this happens, or what this causes overall, is the fact that these guys are inherently difficult to both isolate and purify, and even more so to crystallize, okay? So if we want actual structural determination, we have to form crystals. We have to undergo three-dimensional X-ray crystallography, etc. But with respect to these membrane proteins, they are inherently difficult to crystallize, and so we don't have complete structures for all of them. So a lot of this is just us saying, well, we have this complex from bacteria, or we have an overall idea of what's happening with this complex, but we don't actually have the three-dimensional structure, okay? So we're going to utilize this complex 1 from this bacteria, Thermus thermophilus, to get an idea of how complex 1 works with respect to all organisms, okay? All right, so now, overall, we have 2 different components to this core subunit construction, all right? This guy forms what we can consider an L shape with respect to the plasma membrane, right? So we have one arm of this L shape that lies in the membrane itself, and then one arm of this that actually projects above the membrane. So what we talked about last time was the overall construction of this electron transport chain with respect to mitochondria, right? So with respect to mitochondria, where were these guys located? Where were all of these complexes located? They were located on the inner mitochondrial membrane, right? So that was essentially the dividing line between the mitochondrial matrix and the intermembrane space, okay? Similar things occur in bacteria, except we don't have mitochondria in bacteria. Okay? So instead, what we have is the cytoplasm and the periplasm. The cytoplasm is analogous to the matrix of the mitochondria. Okay? So anytime we have cytoplasm listed in these three-dimensional structures, that is essentially just the matrix of the mitochondria. And then the periplasm is the intermembrane space, or what you can consider the equivalent to the intermembrane space. Okay, so 2 different constructions. We've got our little L shape, or our arm that lies in the membrane, and we have our arm that projects into the matrix, okay? Now, these 2 arms are going to be responsible for 2 separate things. The one that projects into the matrix is going to be our electron transport arm, and the one that lies in the membrane is going to be responsible for translocating protons. So this is going to be our proton transporter. Okay, now we're going to start by talking about the electron transport arm of complex 1. So again, we have our little reactant, our substrate of complex 1, which is NADH, right? Reduced coenzyme coming along. It's going to come up to the matrix side of complex 1, okay? The first thing that it's going to interact with is a flavin, right? So we have a flavin first. And then, as we move down the length of this arm, we have this long chain of iron-sulfur clusters, right? Long chain of iron-sulfur clusters until we get to the very bottom where we actually find our coenzyme Q, which is going to get reduced. Now, why would we need to encounter a flavin first? Thinking about the electron transport properties of these various redox active cofactors, NADH, how many electrons does it transfer at a time? 2. It has to be 2, right? We talked about these iron-sulfur clusters last time. How many electrons are they capable of transferring at a time? 1, right? So we have to have some sort of conduit between a 2-electron carrier and a 1-electron carrier that's capable of donating both 1 and 2 electrons at a time. And that ends up being flavin in this case, okay? So flavins are stable in both the 1-electron and the 2-electron reduced forms. So the first thing that's going to happen is NADH is going to transfer 2 electrons to flavin, forming the fully reduced FMN. then that FMN is going to transfer its electrons one at a time down this chain. Okay, so one at a time down this iron sulfur cluster chain until we reach the bottom where we get to quinone. Is quinone capable of accepting one electron at a time as well? Yes, right? So quinone is going to get one electron reduced first, then finally it will get two electron reduced to form its completely reduced ubiquinol. Okay? Okay, so that is the electron transport side. We're going to talk about the proton transport in a second. So one thing that I should note, and I think I was supposed to do this on this slide, but I've already done it on the previous slide, so it doesn't matter. One thing I should note here is that all of these iron sulfur clusters that form this little kind of chain or this little wire down this complex 1, they are all embedded in the membrane, right? So these iron sulfur clusters are ligated to the protein. They're covalently attached via those cysteine thiolates, right? Those cysteine thiolates were forming ligands to the iron, and that was essentially keeping that iron sulfur cluster in the actual protein itself, okay? Now, all of these electron transports, or all of these electron transfers, are redox reactions, right? And in chemistry, overall, one of the big ideas that we're trying to get across to you is that for things to actually react, they have to collide, right? So if we want to transfer an electron, we have to have our two reactants. They have to collide together. We'll get electron transfer, right? What's happening here, though? If all of these little iron sulfur clusters are embedded in the membrane, they're not moving, right? They're covalently attached to the enzyme. How are we getting electron transfer when these guys are actually fairly far away from one another? So that's a big question, right? And that disturbed scientists for quite some time. What we know now at this point in time is to explain this overall electron transfer, we have to invoke a quantum mechanical theory called tunneling. Okay, so the means by which we actually see electron transfer and all of these various redox chains is via tunneling. Okay, I'm not going to go into what tunneling means, but what you should know about it is that tunneling allows for fast and efficient electron transfer up to 14 angstroms distance with respect to one another. Okay, again, this is a quantum mechanical idea, it's a quantum mechanical theory, and it's going to explain why we see this electron transfer. What's important to note, though, is that if we get larger than 14 angstroms away, we're not going to be able to efficiently and quickly transfer electrons. And so what you will see with respect to these electron transfer chains is that these redox active cofactors do not move farther than 14 angstroms away from one another, because if they did, then the entire chain would be broken, and we'd have to find another species to go about doing this, okay? Okay, so overall, with respect to going from our NADH to our coenzyme Q, down our little arm of complex 1, we transverse about 140 angstroms, and again, that is accomplished via the action of all of these iron sulfur clusters. Okay, so that's great. That is the electron transfer properties of complex 1. Now, what complex 1 is also going to have accompanied with it in this electron transfer is proton translocation from the matrix to the intermembrane space, okay? So again, the cytoplasm is the mitochondrial matrix, and the periplasm is the intermembrane space. So electron transfer occurs down this arm of complex 1. We're also going to see proton movement from the matrix to the intermembrane space via this arm of complex 1, okay? And net, what we're going to see overall is the translocation of 4 protons from the matrix to the intermembrane space. Now, you may say to yourself, how does electron transfer down this arm generate the ability to transfer protons, or the need or necessity to transfer protons distances that are about 200 angstroms away from it? All right, so it's electron transfer down this chain that takes these protons and actually translocates them through the membrane, right? I'm not going to go through the ideas or the necessary mechanisms behind this, but I want to say this. So number one, the big hypothesis behind this is that when this quinone gets reduced, right, it picks up 2 electrons. And they think that when it picks up those 2 electrons, it does not immediately become protonated, okay? So the idea behind this is that it's not protonated until it moves into the membrane. When it does, it's still in this negative 2 charge. As it moves closer to this little component, our proton component, they think that there is a protein structural change that is accomplished via this negative charge on the quinone. And that structural change essentially shifts the ionization, or the pKa's, of a bunch of these residues that are in this membrane-bound portion, so that we get translocation overall of these protons to the intermembrane space. And again, there are 4 of them overall that are moved via this mechanism. Okay? So 2 electrons are going to generate translocation of 4 protons in complex 1. Okay, so that is complex 1. That is what you need to know about it. Who comes next? Probably complex 2. Okay, so complex 2 we have actually already talked about before. So complex 2 is the other component to the succinate dehydrogenase that we talked about in the citric acid cycle, right? So what did succinate dehydrogenase do? It took succinate, and in the citric acid cycle, what did it turn it into? Fumarate. You will know this, hopefully next Thursday. Takes succinate, turns it into fumarate, and in the process, what does it create? It oxidizes succinate. Right, so we get the production of FADH2, okay? That is the first half of what happens in complex 1. The second half is what happens to that FADH2, okay? All right, I also had mentioned last time that in complex 2, taking FADH2 and producing reduced ubiquinol, which is what's going to happen in complex 2, there is not enough energy in this overall reaction to actually get the translocation of protons, okay? So the only thing that's going to happen in complex 2 is we're going to take our FADH2 and we're going to produce reduced ubiquinol, okay? We're not going to get any type of protons translocated because there's not actually enough energy in this overall reaction, okay? Okay, so what are the important cofactors with respect to complex 2? So we're going to have our FADH, and it is going to bind to the matrix side. So we're on the matrix side of our complex 2. Again, this is a 2-electron transfer, or no, it can be a 1 and a 2-electron transfer protein. The first thing that's going to happen is it's going to transfer its electrons to a 2-iron 2-sulfur cluster, then to a 4-iron 4-sulfur cluster, then to a 3-iron 4-sulfur cluster, and finally, it will transfer its electrons to coenzyme Q as well, okay? Again, it does this one at a time until we finally get completely reduced coenzyme Q, or ubiquinol, another name. There is also a heme that is in this protein, right? This heme never becomes formally reduced. They think its presence there is just to modulate the electronic properties of this overall redox chain in coming from FADH2 to coenzyme Q. Okay, so this guy never gets reduced, even though it actually is present in complex 2. Okay, so again, purpose of complex 2, we take the electrons that were present in our FADH2 from the citric acid cycle, and we also produce reduced coenzyme Q. So now reduced coenzyme Q is going to move on to complex 3. So now we have 2 means of getting it there. We have complex 1 as well as complex 2. Okay? Okay, so now we have complex 3. So what's going to happen at complex 3? Right. So we're going to take these electrons that are now in the presence of ubiquinol, right, and we're going to transfer them long-term to cytochrome C. Okay? So complex 3 is responsible for taking our coenzyme Q and transferring those electrons to cytochrome C. Just as a note, how many electrons does this reduced coenzyme Q have? 2 at this point in time, right? So we need 2 electrons for their full reduction of coenzyme Q. How many electrons can cytochromes hold? Just 1. Cytochromes, hemes can only hold 1 until I tell you differently. And so because of that, we're going to get the production of 2 cytochrome Cs for every 1 ubiquinol that enters complex 3. Okay? Okay, so complex 3 is this nice big protein as well. Again, it has multiple different names, so it's also called coenzyme Q, cytochrome C oxidoreductase, or more importantly, cytochrome BC1. This is what it's really known with respect to the scientific community, cytochrome BC1. Okay, so a couple things about the overall structure of complex 3. It is a homodimer, right? So it essentially has symmetry right down the middle of it. What happens on one dimeric or what happens on one monomer is going to happen exactly the same way on the other one, okay? There are also two different redox paths or two different electron transfer paths that occur in complex 3. The first one is going to involve a 2-iron, 2-sulfur cluster. That's going to transfer electrons to what is called heme C1, which is going to directly transfer electrons to our soluble cytochrome C. The other path is going to go the opposite way. We're going to take an electron from quinol, and it's going to be transferred to a cytochrome Bl, which is then going to be transferred to a cytochrome Bh. I'm going to talk about the different pathways that these guys can take and how that factors into the overall mechanism behind complex 3. Okay? But note, there are a lot of different redox active cofactors. One branch has cytochromes, the other branch has an iron-sulfur cluster, and then a cytochrome, which finally reduces cytochrome C. Okay, all right, so let's talk about the actual mechanism behind complex 3. Okay, so in complex 3, we're going to have two separate binding sites. Okay, so two separate binding sites that are going to be called Q0 and Qi. Okay, so Q0 and Qi. Q0 is going to be right up here. So we're going to note it Q0 right up here, and Qi is going to be down here. Quinones can bind to both of these sites. Okay, so one thing to note about ubiquinone and ubiquinol is that it is lipophilic. Right, so that means that it loves the membrane, it loves to be in the lipid membrane, and so because of that, ubiquinone in its reduced form and its oxidized form is going to reside in the actual membrane itself. Right, it's going to be soluble and it'll diffuse freely through the membrane. It's not going to move to the matrix. It's not going to move to the intermembrane space. So we're going to have reduced ubiquinol coming from both complex 1 and complex 2. The first little cycle in this Q cycle, which is going to be what the overall mechanism for this is called, we're going to have our reduced ubiquinol bind to the Q0 site. Okay, so that's going to be number 1. We're going to have binding of ubiquinol to Q0. We're then going to get something called an electron bifurcation. Right? So an electron bifurcation, we have two electrons that are present on ubiquinol. Right? One of those electrons is going to go one path, and the other electron is going to go a completely different path. And that's why we have two separate redox paths within complex 3. Okay? So what's going to happen first? We have binding at our Q0 site. The first electron that is going to be moved, or is going to be transferred, is going to go to our iron-sulfur protein, our 2-iron, 2-sulfur cluster. So I'll say 2Fe, 2S. That one electron is then going to be passed to cytochrome C1, which is then responsible for reducing cytochrome C. Okay, and cytochrome C is the long-term product of this overall cycle. The other electron, so that was just one electron that we moved, the other electron is going to go first to cytochrome Bl. The L here stands for low potential. It is then going to be transferred to cytochrome BH. The H stands for high potential, right? So the second electron is going to go from the semiquinone to Bl to BH. At this point in time, we have fully oxidized the quinone, or the quinol to quinone, right? What's going to happen is it's going to migrate through the membrane, and it's going to bind at the second binding site in its oxidized form, okay? So the second binding site is Qi. Okay? At that point in time, that electron that is now on cytochrome BH is going to be transferred to our oxidized quinone to form the semiquinone radical. Okay? All right, so that's one cycle. Second cycle, same exact thing is going to happen. We're going to have our ubiquinol coming from wherever it's coming from. It's going to move in and bind to Q naught. We're going to get the exact same electron bifurcation, right? So we're going to get movement from one electron to the iron-sulfur cluster, and then to cytochrome C1, which then transfers to cytochrome C, which is our second cytochrome C that we have produced. The other electron is going to take the exact same path. It's going to move down the cytochrome BL, the cytochrome BH, and now it's going to be transferred to our semiquinone to get the fully reduced quinol, okay? So overall, what we have done is we have taken 2 of these guys, and we have produced how many? For this net Q cycle, we take 2, we oxidize 2 of them, but in the process, we also produce 1 over here, okay? So when we take 2, we will get 1 back out. Net for 2 different cycles is going to be the actual oxidation of 1 of our ubiquinols, okay? Okay, so Q cycle, 2 of them go in, we get 1 back out, but overall net oxidation, we have the oxidation of 1 ubiquinol. A couple of things. Complex 3 is going to be capable of proton transfer. It's not going to occur in the exact same mechanism that complex 1 did, right? Complex 1 had electron transfer, which resulted in the translocation of protons via conformational changes. What's going to happen here is chemical. It's purely chemical, right? When we oxidize the reduced quinone, how many protons do we produce? It has 2 protons on it when it's reduced. How many come off when we oxidize it? 2, right? So we get the production of 2 from our first cycle. Second cycle, exact same thing. We get the movement of 2 to the intermembrane space. On the other hand, we take up 2 when we reduce it over here in the QI site, or the QI site, right? So net, we push 2 across, but we take up 2 from the matrix. So net overall, we get the movement of 2 protons in complex 3. Okay, all right. Yeah. Q cycle, great. Again, 2 cycles. You're going to have complete oxidation of 2 quinols, but you're also going to get the production of 1, right? So overall net, we get the oxidation of 1 quinol, and we get the translocation of 2 protons in a chemical manner, right? We call this a chemical proton translocation as opposed to the actual pumping, which we saw in complex 1. All right, so now at the end of this, we have taken our ubiquinone, we have transferred those electrons to cytochrome C, okay? And what we're going to come out of this, for every 1 coenzyme Q, for every 1 reduced coenzyme Q, we're going to get the production of 2 cytochrome Cs. They are now going to move on to complex 4, okay? So complex 4, we're going to take our nice little cytochrome Cs, and what do you think we're going to do with it? Because we're at the very end of our chain. What is the overall purpose of the electron transport chain? It's going to be the reduction of oxygen to water, right? So that's what's going to happen in our final little complex, complex 4. Okay, so before we talk about the cofactors and the mechanism behind this, let's just think about the chemistry of this reaction, right? In this reaction, we are going to have oxygen, and we are going to reduce it to water. How many electrons do we need to reduce it to water? Complete reduction. If I had 1 O, then it would be 2 electrons. 4 overall, right? So complete reduction of an oxygen molecule to water is going to result in 4 electrons. We're also going to get 4 protons taken up, and we're going to produce 2 waters in the process, okay? So before we can do anything, we need 4 electrons. And these cytochromes only hold 1 electron at a time, okay? So we're going to need overall 4 cytochromes coming to complex 4 to actually get this overall process, okay? Another thing to note is the other name for complex 4. This is called cytochrome C oxidase. Okay, so where is cytochrome C? I didn't mention that. So cytochrome C is produced at the intermembrane side, right? So the intermembrane space. So this is where our little cytochrome C is coming from. We're going to get oxidation, or we're going to get reduction of oxygen to water at the matrix side. Cytochrome C is in the intermembrane space. It migrates over because it's nice and soluble. It complex, it comes in contact with complex 4, we're actually going to get these transfers of electrons. So now, how does this occur? So complex 4 has very, very, very complex chemistry, and we're going to talk about that. I'm not going to actually go into the real overall mechanism, but I'm at least going to tell you where these electrons come from when we reduce oxygen to water. Okay? So there are 4 central redox centers in complex 4. The first one is called the copper A center. Okay, so this copper A center has 2 copper ions, so 2 copper ions. They are ligated to the protein in a very similar method or manner with respect to an iron-sulfur cluster, right? So they have ligands that come from our side chains of our amino acids. These are histidines, methionines, etc. And they are also coordinated by inorganic sulfides. So it's very similar, very reminiscent to an iron-sulfur cluster, except it has copper instead of iron. That's first center. Copper A center, binuclear, has 2 copper ions. Second center, we call heme A. Again, this is just a heme, this is just an A-type heme. Third center is called heme A3, but most importantly, it is in very close proximity to something called copper B. Okay? Copper B is just 1 copper atom, but because it's so close to this heme A3, essentially forms a binuclear cluster with this heme A3, right? So hemes have ions in them, copper has copper, right? These guys are really close together. This is going to be the actual active site where we reduce oxygen to water, okay? So what's going to happen is we're going to have cytochrome C. It's going to transfer 1 electron at a time down our little redox chain in complex 4. The first site is going to be this copper A center. It's then going to transfer an electron to heme A. It's then going to transfer an electron to heme A3 and copper B. Okay? That's the mechanism of the method behind this electron transfer. Now, at this binuclear heme A3 copper B center, this is where oxygen is going to bind, right? This is where we're actually going to get the reduction of oxygen to water. Again, how many electrons do we need for that process, that overall reduction of oxygen to water? 4. We need 4, right? Now let's think about the oxidation states of these 2 metals with respect to this binuclear heme A3 copper B center, okay? So iron, what is the fully reduced version of iron usually found in biology? I'm not saying fully reduced iron metals, I'm just saying oxidation states. So yeah, fully reduced iron is iron 2+, right? So it has a 2+ oxidation state. The fully reduced version of copper is copper 1+, right? It's in the 1+ oxidation state, okay? So this center is initially fully reduced, okay? What we're going to see is one of these electrons, because we need 4 overall, one of them is going to come from copper, right? When we oxidize copper with 1 electron, that is going to produce copper 2+. So 1 electron comes from copper, 1 electron comes here. 2 electrons are going to come from the iron in heme A3. Okay, so iron in heme A3, we're going to lose 2 electrons from that. That's going to form this iron 4+ oxidation state, right? This is unusual, but this is pretty dramatic chemistry anyway, so we're going to have to invoke some unusual types of characteristics. So this iron in the 4+ oxidation state, this is called ferro iron. Okay, so that's 3 of them, right? Where does the 4th electron come from? There's no more metals around to give up electrons. So instead, again, in invoking this very weird chemistry, what's going to happen is this 4th electron is going to come from one of our amino acids, one of the side chains of the amino acids that actually line the active site. And that side chain is the side chain of tyrosine, right? So what is the side chain of tyrosine? It's not just an OH, right? It's a phenol, right? It is a benzene ring with an alcohol, right? The presence of that benzene ring, what does it make this alcohol comparatively just a normal alcohol with respect to chemistry? Much more acidic, right? So in the active form of this is going to be deprotonated, okay? So if we're deprotonated, then we already have a -1. This is the phenolate ion. What's then going to happen is this thing is going to lose an electron, right? Just one electron. If you lose one electron, what do you form? A radical, right? So we get the promotion or the formation in this process of a tyrosine radical, which is odd and unusual, but this is what has been shown to occur, and this is actually utilized in other mechanisms and other more complex chemistries in biology. Okay, so we have 2 electrons coming from the heme A3, we have 1 electron coming from copper, and that last electron is going to come from this tyrosine residue, and by doing it, it's going to promote, or it's going to form this tyrosine radical. Okay. All right, so those are the 4 electrons. We're going to get oxygen binding. We're going to get the complete reduction of oxygen to water. We get 2 waters out of it, okay? Now, this process is also going to translocate protons, and it's going to do so in a proton pumping mechanism similar to what we saw in complex 1, okay? So we're going to get the pumping of protons from the matrix to the intermembrane space. That specifically ends up being 4 protons pumped, okay, for every 1 oxygen forming 2 waters. If you think about the chemistry of this, though, so this guy out here is, again, our intermembrane space. This guy is our matrix. What else occurs with respect to the reduction of oxygen to water in terms of protons? So when we reduce oxygen, do we utilize protons in that actual transfer or that actual chemical reaction? O goes to form H2O, right? So this process also takes up 2 protons from the matrix, right? So we get the taking up of 4 protons overall when we take oxygen and we reduce it to water, right? So that is also going to contribute overall to our little electrochemical gradient that we have been continuously making as we've gone through this redox process, okay? So not only are we going to get the translocation via proton pumping mechanism similar to what we saw in complex 1, we're also going to get this chemical mechanism by which we take up protons when we form water. Okay, so those are the active cofactors, all the complexes of complex 1. So really quickly, before we get into oxidative phosphorylation, let's try a Socrative question. Just make sure you guys are paying attention. Okay, so go ahead. Most of you got your answers in. If you haven't, quickly do so. 86%. So that's great. So what is the correct answer here? A. Should have made it later down in the list, so you have to actually read the other ones, but that's fine, because I have another. Let's try this one. I want to know which of these statements is false about the electron transport chain. Okay, so go ahead and get your answers in if you haven't. What is the correct answer here? Which one is false? It is C. So it says complex 3, also called succinate dehydrogenase, is shared with the citric acid cycle. Which is the correct answer here? This is complex 2. So we wanted to change this to actually be correct. It is complex 2 that is shared with the citric acid cycle. Okay, all right. So one more thing that we have to talk about. We've gotten NADH, we've reduced oxygen to water, we've generated this proton gradient, right? So now, that has to be coupled somehow with the synthesis of ATP. And I'm sure you guys already know how that is coupled, right? So we have one more little thing that we have to talk about, just how we're going to use that proton gradient that we have generated to synthesize ATP. Okay, so let's keep track a little bit of what we have generated thus far, okay? So for one cycle of this, when we come into complex 1, we have NADH. So NADH in complex 1, how many protons does it translocate from the matrix to the intermembrane space? For complex 1, we got the net movement of 4. 4 protons in complex 1, okay? Complex 1 are those electrons from NADH turned into ubiquinol, right? They moved on to complex 3. How many net protons for every reduced ubiquinol did we get through complex 3? This was the Q cycle. For every 1 ubiquinol, we got the translocation of 2 protons in complex 3. And then from complex 4, for every 2 cytochrome Cs, how many protons did we get through complex 4? Net. Net, we will get 4 protons overall. Okay, so we'll just clarify that. We have 4 from complex 1, 2 from complex 2, 4 from complex 4. And again, this is from 1 NADH, okay? So how many net protons is that overall for every 1 NADH moving through this electron transport chain? That is 10. So for every NADH, we will get 10 protons. What about FADH2? 2 as well as M2. Where does FADH2 enter its electrons? It enters in the second one, but we don't get any type of net movement of protons in complex 2. So FADH2 is limited to the production of protons in complex 3 as well as complex 4. So for FADH2, how many net protons do we get moved across the membrane? 6. 6 protons total. Okay? Okay, so that's just keeping track. Now, what we have generated is called a proton gradient, right? So we have made a net movement of protons from the matrix to the intermembrane space, okay? This is going to generate 2 different things, right? So first off, we have a chemical gradient, right? We have a high concentration of a chemical, protons, on the intermembrane space comparatively to the matrix, right? So what do things like to tend to do with respect to high concentrations versus low concentrations? Diffuse back down, right? So that's the chemical component of this proton gradient. There is also an electrochemical component, right? Not only is proton a chemical, but it's also a charged molecule. So as we have been pumping these protons out, we have generated a bunch of positive charge in the intermembrane space comparatively to the lack of positive charge in the matrix, okay? So this electrochemical proton gradient has 2 different components. Net overall, to translocate 1 proton from the matrix to the intermembrane space, it's going to cost us about 21 kilojoules per mole of energy, okay? So the net transport of 1 proton to the matrix costs about 21 kilojoules per mole. Now, there are 2 different components to that. Again, one is the chemical and one is the electrochemical. The chemical component does not actually contribute all that much. It only contributes about 4 to 5 kilojoules per mole to this overall net gradient. On the other hand, the electrochemical component is really where all of this energy comes from. It generates the rest of the energy, which is about 15 kilojoules per mole. And again, this is the energy required to move a proton from the matrix to the intermembrane space. Now, if we think back to when we originally started talking about this, and we calculated the energy that was going to be released in taking NADH and reducing oxygen, what did we calculate? How much energy was available in that overall reaction? So delta G overall from NADH, taking those electrons and producing water, that gave us about minus 200 kilojoules per mole, right? So that's how much energy is available in just the net movement of electrons from NADH to oxygen to produce water, okay? How many protons do we translocate from 1 NADH? 10, right? So we get 10 translocated across the membrane. About how much energy do we use in doing that? If it costs 21 for every 1. About 210, right? So it's a fairly efficient process. We had 220 kilojoules. We used 210 of it to get those 10 protons across, okay? So now, how are we going to couple this to ATP synthesis, right? So for a very long time, people thought that these electron transfers and all of these various complexes were generating some sort of high energy intermediate, right? Just like we see in glycolysis. So in glycolysis, how do we get ATP synthesis? We produce molecules that have higher energy of hydrolysis than ATP itself, right? We produce 1,3-bisphosphoglycerate, right? That was capable of phosphorylating ADP. We also produce phosphoenolpyruvate. What is that called? That process of taking something and directly phosphorylating ADP? Substrate level phosphorylation, right? So they thought for a long time that these 3 complexes that had enough energy to generate proton translocation were also making some sort of high energy intermediate that was directly producing ATP, okay, via substrate level phosphorylation. Now, we know at this point in time that that is not correct, right? Instead, what is generating this ATP is essentially allowing this proton gradient that we have promoted or that we have formed in our electron transfer to come back, right? To disperse back through the membrane. If it costs 21 kilojoules per mole of energy to get a proton out, how much energy are we going to get when we allow it to come back in? Just the reverse of that, right? Minus 21, right? So if we have endogonically pumped the proton out, if we want to release that, it's got to come back in, and we're going to get that energy back. That's going to come in the form of complex 5, which is ATP synthase. And this coupling of this electron transport to this proton gradient to the actual synthesis of ATP is what is called the chemiosmotic theory. Okay, this is what is governing this overall idea behind oxidative phosphorylation. Okay, so what are a couple of the things that have led us to believe that this chemiosmotic theory is correct? Okay, so there are a couple of key observations in this. Number one, big key observation is that inner mitochondrial membrane is completely impermeable on its own to charged molecules like protons, as well as potassium and other types of charged molecules, right? So that's a big one. If you do something that makes that membrane permeable to these types of charged molecules, what you will get is still electron transfer through the electron transport chain. You will still get the reduction of oxygen to water, but that will be uncoupled to ATP synthesis, right? So there has to be something in the generation of this gradient that when you release that gradient without actually allowing it to come back in whatever source it's going to go through, it reduces this ATP synthesis overall. Okay, so important things is this impermeability of the inner membrane, and also the fact that you can uncouple this, still get electron transfer, but not actually get ATP synthesis if you allow these molecules, these protons and these potassium ions, to enter the membrane again through some other means. Okay, so great. Now, how is it actually going to be coupled, right? So we're going to talk about this guy, ATP synthase, or complex 5. Okay, so ATP synthase has lots and lots and lots of different names again. So we call this F1 F0 ATPase. This is also called complex 5, as well as ATP synthase. Okay? All right, so there are two components to this overall complex. There is what is called F1, and there's what is called F0, or what I like to call F0. Okay? So F1 is present on the matrix side of that inner mitochondrial membrane, right? So F1 is water soluble, and it's present in the matrix. F0 is not water soluble. It is embedded in the membrane, this inner membrane, and this is what we get right here, okay? So a couple of things. F1 can be removed from F0, right? You can disrupt it via sonication. You can get F1 on its own by itself. It is not capable of synthesizing ATP, but it is an ATP hydrolyzer, meaning it can take ATP and hydrolyze that bond and make ADP and PI. That's why we call this an ATPase, right? A synthase is going to make ATP, and ACE is going to hydrolyze ATP, okay? So if we remove F1 from F0, we will get hydrolysis spontaneously, right? It can't synthesize it, though. A couple of the other things, so we have a couple different components to this. We tend to call this a lollipop-like structure, right? It has two components to it. It has this big spherical head that is connected to F0 via this little stalk. Okay, so we're going to talk a little bit about what this stalk does. But again, overall, it kind of looks like a lollipop embedded in the membrane. And just as a reminder, this is our intermembrane space, and this is our matrix. That's where F1 actually lies. Okay, so now let's take these guys apart for 1 second, okay? So we're going to talk about the F1 unit first. Again, this is our matrix, little lollipop-like shape, okay? So there are a couple different subunits that go into the overall construction of F1. Particularly, the ones that we're going to be interested in is this alpha 3, beta 3 construction, as well as this little subunit gamma. I'm going to explain what each of these are as we go through this diagram. All right, so alpha and beta form our overall monomeric unit for F1. Okay, so alpha beta form this monomeric unit. There are 3 of them overall. So with respect to this alpha-beta monomeric unit, they essentially form a trimer, and that's what forms this whole spherical head. Okay, so if we look at downward at this thing, we'll find an alpha-beta unit here, we'll find an alpha-beta unit here, and we'll find an alpha-beta unit here. Okay, so again, one of these is our monomer. Together, they form a trimer. Okay? Also, another thing to note, alpha and beta are very similar with respect to one another. They share a lot of similar structural similarities. That's what I meant by that. Lots of structural similarity. They both are capable of binding ATP. So they both have ATP binding sites within the two of them. Hydrolysis or synthesis of ATP is only going to occur at the beta subunit though. So even though alpha can bind ATP, we don't actually get any type of synthesis at alpha. Okay, so how do these guys differ with respect to their little trimeric unit? So there are 3 different structural conformations of this alpha-beta depending on the presence of somebody else, okay? So one of these structural units is called DP. So in the crystal structure of this overall F1, DP has ADP and PI bound to it, right? So in the crystal structure, in the structural form of DP, we find ADP and PI bound to the beta subunit. Another word for this, or another terminology that we use for the DP overall, is what is called the loose state. So L is what we abbreviate with respect to DP. Another structural conformation with respect to this alpha-beta monomer is what we call the empty state, right? So when they actually characterized this structurally, they found that the beta in this guy was empty, didn't have anything in it, okay? We also call this the open state, and I'll show you what I mean by that in a second. And then the final conformation of this is called the beta TP state. In the overall structural of this, we find ATP bound in beta in the TP state. And what we term this guy is the tight state, or T. Okay, so now, how do these alpha-beta units actually differ with respect to one another? Or what's going to determine that? They're all the same protein, right? Alpha-beta here versus alpha-beta here versus alpha-beta here, same exact thing, right? So how could they structurally differ from one another if we do actually have structural differences between this guy versus this guy versus this guy? Because one of them has ADP, one of them is empty, one of them has ATP. What could be essentially causing that? Conformational change, but it's not going to be in those proteins itself, or it's not going to be caused by those proteins itself. There's going to be another protein that's going to overall lead to these conformational changes, and it's this guy right here. This is what we call gamma, okay? So this thing kind of goes up into the head of the lollipop. This is our stalk. This is what we call gamma. Gamma is going to rotate. That's the big overarching story of this. Gamma is also asymmetric. So when it rotates, its motion is going to actually cause structural conformational changes in each of these various monomers with respect to alpha beta. That's what's going to lead to it either being ADP, ATP, or empty. Okay? So gamma is going to rotate. Overall, the alpha beta trimer is going to stay the exact same, but we are going to see structural conformational changes inside of alpha beta because of rotation of gamma. Okay, so now gamma rotates, right? But who is gamma attached to overall? So gamma is attached to F naught. And most importantly, gamma is attached to the C subunit of F naught. So F naught, as a reminder, is embedded in the membrane, okay? It has a couple of different subunits that are going to be important. Of course, in moving from F1, we had Greek alphabet. When we go to F naught, we have our normal little alphabet. So the overall subunit composition is A, B, and C, okay? Again, these C's are what are actually attached to our gamma subunit of F1. So now, what is the C? What is it actually doing? So this guy is a transmembrane complex, okay? And it consists of a couple of different variations of the number of C subunits. Overall, C is just this two helical thing, right? So we have multiples of these that are stacked together to form this overall C bundle that sits in the membrane, okay? But again, overall C by itself, 1C, is just an alpha helical, 2 alpha helices. Okay, so another important thing after C is A. A is right next to C. And it looks, when we actually look at it structurally, like a soda pop, right? Just like a can that sits in the membrane. Nobody got that. No, okay, so again, this is one of those things that we actually don't know the structure for, right? We have the structure for the C subunits. We don't know what the actual A subunits look like, but we think it approximately looks like a helical bundle that sits in the membrane, kind of like a little soda can, okay? Other things to note are the B's. The big thing about the B's is that, number one, they are tethered to the membrane, but they reach out and over, and what they essentially do is they hold that F1 in place, right? So those alpha beta units of F1 are held in place and are not allowed to move because of the tethering of this B subunit. So overall, what's going to happen, alpha beta doesn't move, gamma does. Gamma is moving because we also see movement in the C subunits down in F naught, okay? Okay, so how is that actually going to lead overall to ATP synthesis? So for this, we induce what is called a binding change mechanism. This was introduced by Paul Boyer in the 1970s. So what happens here is, again, we have our nice little ATP synthase, our F1 up top. It's got those little alpha beta dimers, or alpha beta monomers that form this trimeric unit overall, right? And they are all structurally different depending on where gamma is, okay? So when we start this binding change mechanism, what we're going to start with is we're going to start with open in our loose form, or we're going to start open in our loose form, also open in our open form, and we're going to have ATP bound in our tight form, okay? So now, ADP and PI can only bind to the L version of alpha beta, right? So ADP and PI are going to come in and bind in the L region, okay? At that point in time, we're going to get rotation of gamma. Gamma is going to rotate 120 degrees, right? Upon rotating, it's going to change the conformations of everybody, right? So what was open is now going to be loose. What was tight is now going to be open, and what was loose is now going to be tight, okay? Upon doing that, whenever this guy changes into the open, ATP can be released. So ATP can only be released in the open version of this overall mechanism. Also, what's going to happen is that hydrolysis occurs when we're in the tight conformation, right? So we had ADP and PI bind, we changed it into the tight, and so that's going to structurally make those two come together, and we're going to get the synthesis of ATP in the tight, okay? At that point in time, again, we have our nice loose form, it's open, it's ready to bind ADP and PI. We're going to get binding, and we're going to get rotation of the gamma again, so that everyone changes overall again, okay? So full rotation, going from here and then long term back to here, is going to produce 3 ATP overall. So full rotation of our ATP synthesis or synthase produces 3 ATP overall. Okay, all right, great, right? Awesome. So we get this rotation, that's awesome, that's neat, but how is that coupled to protons moving across the membrane? I don't know, unfortunately. No, I do know. Some people kind of have an idea about how this is coupled. We're going to get rotation, right? That's what's going to produce ATP. But how is that going to be coupled with protons that are on the intermembrane space, coming back across the membrane, rotating this somehow, and producing this overall ATP long term, okay? I'm going to try to explain that as succinctly as I possibly can. Okay, so the big overall idea, the big overall hypothesis about how proton translocation turns gamma, which produces ATP, is as follows, okay? A, right? So in F naught and F0, which is these guys that lie in the membrane, we had C's and we had A's, okay? We don't know the structure of A, but what is hypothesized is that A has 2 separate hydrophilic channels, okay? One hydrophilic channel is on the intermembrane side, where all of the protons are, right? And another hydrophilic channel is along the matrix side, right, where the protons are going to go to as they move through F naught. Okay, so the idea behind this is that in this hydrophilic channel that's on the membrane side, we're going to get protons moving up into this hydrophilic channel, and when they move up, they are going to protonate a residue on these C helices, right? So we had multiple helices that made up this big transmembrane region of the C subunit. We're going to get protonation of one of these helices, and that is going to cause rotation, okay? And I'm going to talk about how that causes rotation in a second. So we get protonation, we get rotation. That's going to open up that exact same residue on another C helix. We're going to get protonation, we're going to get rotation, right? So we keep doing that until these guys long term end up over here. And over here, there is another hydrophilic channel where the pKa has changed of whatever this ionizable residue is. We're going to get deprotonation, and that means it's going to move into the matrix space, okay? So we get protonation of the same amino acid residue on the intermembrane space. We move across 180-degree rotation. We get deprotonation on the matrix side, okay? Now, how could the protonation state of a protein make something move? Right? So we're protonating something, but how is that going to make this thing rotate? Because that's the big idea. We have to get this rotation. So unfortunately, it's still hypothesized, but the current prevailing idea lies with respect to these C subunits, okay? So again, these C subunits are these two helices that are attached together. We can isolate these Cs by themselves, right? So this is devoid of all of their other C helices, and we can analyze them via NMR by themselves. So what we find is in the deprotonated state, so the deprotonated state will say high pH, we find the C helix kinked, okay? So when it's deprotonated, it's kinked. When we then protonate it, so we add a proton to it, and that ionizable residue is this aspartate 61. When it becomes protonated, it goes from this kink form to this straight form, right? So when this guy picks up a proton here, it's kinked beforehand. When that aspartate gets protonated, it stands up, right? And it's the pushing off of A that causes this rotation, right? So deprotonated, gets protonated, kinks up and pushes off of A, and that's how we actually see this rotation of the C subunits, okay? When C rotates, that rotates gamma, which causes all of the conformational changes up in alpha and beta to change, to go from open to tight to loose, etc., okay? So again, it's structural. When we get protonation, it's kinked beforehand. When it's protonated, it goes straight, and it's that pushing off of C from A that causes this overall rotation, yeah. What? Sorry. No, so again, it's deprotonating on another side of the protein, right? So the deprotonation doesn't come back to the same channel. And no, it doesn't, yeah. Okay, so that is the overall mechanism by which ATP synthase allows protons to come back through our nice little inner membrane, right? Via that translocation back through, we get rotation of the C subunits, which rotate gamma, which make all those changes in alpha beta occur, right? And we get overall full rotation, 3 ATP, okay? Now, one thing is important. I never talked about how many actual subunits are in this overall C unit, okay? This differs depending on the organism that we're looking at, right? So some organisms have 8 subunits, some organisms have 12, etc. The amount of these guys is actually going to be kind of important, right? Because the amount of C subunits is going to essentially tell us how many protons we need to get full rotation and full production of 3 ATP. Okay, so just keep that in mind. Okay, so then that is almost it, just to keep track of what we have thus far, right? So we started with glucose. In glycolysis, it was fully oxidized to produce 6 CO2. How many reduced cofactors did we get out of that full oxidation from glycolysis to the citric acid cycle? We got 10 NADHs, right? And how many other things did we get? We got 2 FADH2, right? So now we took all of these little guys through the electron transport chain, right? So for every 1 NADH, how many protons did we get overall? 10. 1 NADH got us 10 protons translocated. What about FAD? We got 6 protons translocated, right? So now, eukaryotes have 8 C subunits, right? So 8 C subunits, that's going to determine how many protons are actually needed for full rotation. One full turn is going to, in theory, give us 3 ATP via that binding change mechanism. Okay? In actuality, this ends up being, what is it? Yes, 2.5 ATP. Okay, so one full turn doesn't actually give us a 3 ATP, it gives us about 2.5 just because of the constraints of the system, okay? So 1 NAD should give us 1 full turn, all right? 10 protons, we need 8 to get a full turn. 1 NADH should give us 1 full turn, which gives us about 2.5 ATP. That's where that conversion comes from, okay? Overall, this conversion between reduced cofactor, or more importantly, electrons, and the amount of ATP that is hydrolyzed or made is called the P to O ratio. Okay, so this is phosphate or inorganic phosphate that is taken up in oxygens that are actually reduced. That's what we're looking at when we're looking at how many electrons do we need to make all of the ATPs that we do in oxidative phosphorylation. Okay, so with that in mind, if 1 NAD gives us, or 1 NADH gives us 2.5, how much do you think 1 FADH is going to give us? Because it doesn't give us the full 10 protons. It gives us 2/3 of that. So 2/3 of about 2.5, that's where we get the 1.5 ATP from. Okay? Okay, so I would suggest looking at uncoupling, right? Various uncouplers in the textbook. There are a couple of questions on the homework about that, and I didn't get to it. But that is the only thing I didn't get to. So I feel very successful about this class.